end up here with a talk on active matter. So I think you had already one last week, but I will start with a quite general introduction, a short one about what we call active matter. And then I will go on with some illustration of our work, which intend to use a biological system to answer simple physical questions. So we use a microalga as a model for an active particle. Ah oui, merci. Ce sera mieux. So let's start with the general information. I think you all might have seen this kind of example. We used to date, let's say, the birth of this domain, active matter, at least in physics, with the inspiration that Vicek had by trying to describe with physics a flock of birds. So here what this illustrates is that you can observe collective behavior, emergent pattern, collective behavior with a pattern that can be dense and moving in a system of an assembly of individual entities that are able to move. And Vicek was one of the first to propose a simple physical model for this system with very minimal ingredients, and this led to what we call active matter. So if we're trying to give a simple definition, we used to say that active matter is defined as a system that is able to consume energy and produce some work, that is most of the time composed of self-propelled units, and that the typical, the specificity of those systems is that the drive is exerted on each particle and it's not driven by the boundaries or externally as in most non-equilibrium systems that we know of. And very often that there is, as a consequence of those simple ingredients, emergence of collective behavior, such as flocking, for instance. So as you see in this definition, this is very general and that can apply to many systems in many ranges of sizes, going from subcellular molecular motor, for instance, to crowds of people. And this is only in the living world, but it's, as you can see, a very general definition for many very different systems. As a physicist, we are always trying to find like universality in those systems and extract minimal ingredients that could be applied to such diverse model, or at least define some classes of models for those systems. And the goal for a large community of physicists is to try to reuse the tools that we know of, mainly from condensed matter physics, and to describe this new kind of matter, let's say, and the generic behavior that can be observed in many systems, even though, as we discussed today in several talks, the aim is not to describe quantitatively with one single model all the details of any system. So as an extra definition, an active particle would be a particle that transforms in living systems mainly, most of the time, chemical energy into motion by using a cyclic transformation mechanical that is given by biochemistry, let's say machinery. A typical example will be the bacteria E. coli that use flagella to propel itself using the nutrients in the medium around it. So as I said, there's a different system, and most of the time you can see the emergence of collective behavior in all those scales. So I just draw here several examples in the living world. You can see molecular motors at the intracellular cell on the bottom right, to the fish schools on the up left picture, and through the plankton pictures that you can see here close to Brittany in France, which are the system I would be using in my experiments, as I will show you. Let's say the artificial counterpart or synthetic counterpart are, I just chose 2 examples, but you can imagine many systems where people have tried to build up synthetic active particles that would self-propel by itself, taking up chemical energy the same way, let's say, or in a biomimetic manner, to be able to have a better control of the active particle, not having to rely on biology to control the propulsion and the ingredients. So here are two examples at different sizes. Some bugs robots, centimeter-sized robots on the right, or Janus particles. So those are synthetic particles that have 2 sides that react differently in a solvent, let's say, and can show self-propulsion, spontaneous self-propulsion in a solvent. And on those systems, usually you can observe again self-assembly and collective motion of those systems. So this is like a list of examples, and some I just retain some basic models that we usually use, or we can use to describe active matter. So the simplest model that you would like to use is what we call active Brownian particle, that is able to consider a spherical particle that will self-propel. I will detail a bit the ingredients of this model. Then from this minimal model, you can show that interacting active Brownian particle can lead to collective motion of self-assembly in this system. So this is called motility-induced phase separation, MIPS. Then you can go beyond and add different ingredients in the models. Alignment can be used to drive the interaction between particles, and with alignment, this was historically one of the first models. Vic model would show the emergent, again, phase transition toward a dense moving phase, let's say. And finally, if you take into account the fact that most of the time the active particle in the, let's say, in the bio world are immersed in a liquid, the interaction between the active particle can be mediated by hydrodynamic interactions. So sometimes it's relevant to take into account hydrodynamics to account for some phenomena. So I just detail here what we call active Brownian particle. So you can consider a spherical particle and assume that this particle will have a given velocity that is constant, and you just add a rotational diffusion to this particle. You just take into account the rotational diffusion, and those ingredients are enough to build up the simplest active particle that you can think of. So the equations here are just defining the dynamics of the position of the particle and its orientation. So the orientation is given by a white noise related to rotational diffusion, and the displacement is given by the velocity that we have. And with this, you can come up with, let's say, one of observable that you can have on the right. I don't find my mouse, but on the right, you can measure the mean square displacement of such a particle, and you will find, for a passive Brownian particle, you remember that for V0 equals 0. So when you don't add this this velocity, you will find a linear behavior, whereas for an active particle, you can find this two regimes, a ballistic regime corresponding to this velocity, and at long time scales, you find a diffusive-like behavior, and the different curves represent different V0 in this model. So this is what we call a persistent random walk. So at short time scale, you can define a persistent direction of the particle. And with this simple model, you can go and toward dense suspension and obtain such a result that you can see on the right, moving dense phase of particles. And this we can understand with like pair interaction ingredients. If you have two particles coming side by side in front collision, let's say, to be able to escape this interaction, you have to wait for diffusion, rotational diffusion to give rise to new orientation so that the particle can escape. So if the noise is not too large, you can imagine that a pair will form and a cluster, this is the nucleus of a cluster. Then we can show that this cluster will be moving eventually. So here there's no other ingredients than these active Brownian particles with a high density and let's say not too much noise. Of course, if you shake the system with noise, so you expect to break these kind of patterns. And if I just mention the historical model of Vic-Sheck, so Vic-Sheck is inspired by the bird. For a physicist, a bird is mainly an aligned thing. So this is a rod with a velocity and an orientation, more or less. This means a little bit of noise, or you just tune the noise that you would put in this orientation. And this active particle, you consider that it interacts with its first neighbor. The orientation of the particle, you just assume that this particle will take the mean orientation of its first neighbors, okay? And with not too much noise again, you can observe, this is an example from Vic-Sheck paper, the high density case and low noise case, where you can observe, let's say, a polar ordered phase of a motile cluster of this. And finally, if you go and take into account, if you want to take into account the hydrodynamic interactions and the effect of the solvent on the particles, let's focus on the left side of the top panel. You have a bacteria that is propelling in a solvent. You can model this bacteria as a force dipole hydrodynamic forces. So the tail, the flagella, is exerting a force on the fluid to propel the bacteria, and the fluid is exerting a drag force on the body of the bacteria as a counterforce. So the system is mechanically isolated. The sum of the forces are zero, but with this dipole of hydrodynamic force, you have a self-propelled particle, and you see that the blue arrows indicate the type of flow that this particle will create in the solvent. So this flow will interact with all other particles, and this typically can be long-range interactions in a fluid. And a consequence of this, the two bacteria of panel C, you can show that they would attract if they swim side by side hydrodynamically. On the right side of the panel, it's another system. So it's inspired by Chlamydomonas renarti, the microalga I'm working with. And this you can model as an opposite dipole, because we call this a puller types microswimmer. So this one is exerting a force that pulls on the fluid in front of the body, and the drag force is behind. So the fact that the dipole of force is inverted creates a flow that is opposite, and the interaction between the particles are then modified because of the self-propulsion mechanism that is different. Okay, so this is just to illustrate this kind of modeling, and again, you can show the emergence of collective motion in those systems when you take into account pure hydrodynamic interactions. So as I said at the beginning, the goal is to try to, because the ingredients of the models are so simple, we might think that those models can apply to many, many systems as different as they are, or at least define some class of active particles, and one of them can be what we call microswimmers. So those are defined by micrometer size. So this rules that the hydrodynamic forces are completely driven by viscous forces. There's no inertia in this system, and they're suspended in the liquid. So probably hydrodynamic can be important in those systems as a way of interacting with each other. Okay, and indeed, in many experimental system here, you have mainly synthetic. No, you have also sperm cells in the middle. I will not enter the detail, but in many experimental realization in laboratories of colleagues here, you can see the emergence of collective motion replicated in a lab in a controlled experimental setup. Okay, with this general information, so now I would like to switch to what we do in the lab, and our strategy, let's say, is to instead of trying to build up a synthetic particle, active particle, we let biology do the work. So the cell culture will provide thousands and thousands of active particles in 1 milliliter after waiting for 2-3 days. So we chose Chlamydomonas renarti as a model system. So this has different properties that are interesting, I find, to tackle these kind of questions. It's a quasi-spherical cell. So this is quite nice for the modeling. You can model it as a sphere. It's really swimming in 3 dimensions. So there's many synthetic microswimmers that are living close to the surfaces and interacting a lot with walls. Here you can set up a 3D experiment with this system. Its size is 10 micrometers, so it's large enough so that thermal fluctuations are not relevant for this system. Mainly swimming is important, and the system is not subjected to thermal fluctuations. It, as I said before, it pulls the liquid to propel itself. Okay, so this is something we cannot control, and it's quite easy to culture. And we can also culture it in a certain way to have, let's say, a monodisperse suspension to be able to do physics with it. And with this system, we try to establish some general properties of active particle. So the goal is that if the experiment we set up, if we manage to model them with a model that forget about the specificity of the model, the goal is achieved more or less. And in particular, when relevant, we look at the effect of hydrodynamic in this system. Okay, so here you see what we can see on the microscope for the low magnification movie. You see particles that are, I think by eye, you can see that this doesn't resemble Brownian motion. You can see that there are swimming particle. And as I told you, they use a kind of breaststroke-like motion of the flagella, la brasse in French. But with no inertia. So it's counterintuitive when you're not used to it. I mean, when you swim yourself, I think with just one motion of your arms, you can stop and glide for 10 meters. Me, at least. But for Chlamydomonas, as soon as it stops, there's an instantaneous stopping of the body because there's no inertia. And then the strategy not to go back to the first initial position because the flow is reversible is this special shape of the flagella where the recovery stroke is having less drag than the power stroke. Okay, and this has been shown to fulfill quite well the long range hydrodynamic field corresponding to the force dipole I mentioned before. Okay. So I will try to get through different applications or different examples of experiments just to illustrate the way we try to address this kind of questions, if it's okay. So first, we need to just characterize the microscopic properties, let's say. So we looked at the random walk of this system. So what you see here are trajectories of a suspension. And what we measured, as I showed before, is one of the observables that you can measure is the mean square displacement. What you find is indeed that the mean square displacement is quadratic at short time scale. So this means that you can define a velocity, a mean velocity of swimming for the whole suspension. And at long time scale, you can find a random behavior. So it's a linear behavior. And this can be described by what we call a persistent random walk, which has an expression that connect, let's say, the two limits I'm just talking about, the ballistic regime to the diffusive regime. So here, there's no rotational diffusion or thermal fluctuations. What happens is that okay, the swimmer is having its direction with a given velocity, but there's a kind of noise, intrinsic noise due to biological asymmetry, let's say, that give rise to the change of direction. So you can see it as a noise. You don't need to know the biological origin or thermal origin to model it as an active Brownian particle, as I showed you before. Okay, so the equivalent of rotational diffusion is just a biological noise. Another characteristic of this system, which is something we use to control some experiments, is what we call Phototaxis. When you have a stimulus of light on the side, so the system has the simplest eye of the biological world, apparently, which is not so far from what we have in our eye, except that it's just a photoreceptor at one wavelength. But when a light signal is coming from the right, the biochemistry of the cell will lead to a dissymmetry of the flagella. So one, let's say, will not beat anymore. The other one, yes. So this would lead to a rotation toward the source of light. So this is the mechanism of phototaxis. And this we can characterize. So when there's no light, the system is isotropic. You have trajectories in all directions. You can measure, for instance, the x component of the velocity. If you project the velocity in one direction, you will see that in average is 0. Whereas when you switch on, the light, you see a purely ballistic regime, and you can measure the response, the typical response time of the system to light stimulus. For instance, here it's about 2 seconds for the suspension to react to the light stimulus that you switch on at t equals 0. And you can go a little bit beyond if you want to feed the theoretical models with as many constraints as possible. You don't want to leave any adjustable parameters. If you look at the distribution of orientation of the cells in a suspension toward light, so it's not peaked, as you see in in the trajectories, they are not all exactly directed toward the light, but you can measure the shape of the distribution of orientation, and this shape and width of this distribution you can use in the model to bias the random walk when needed. Okay, so this system, we show that we can characterize its random walk. It's a persistent random walk. We know, so the typical time scale or the velocity is about 100 micrometer per second, which is like 10 body lengths per second. Reorientation time around 1 second, and that we retrieve a diffusion-like behavior at long time scale. And this is, as I said, quite a generic behavior of microswimmer. Whatever the origin of the noise is, you can find very often this type of behavior. So that resemble or that corresponds to active Brownian particles. And the Phototaxis response we characterize, and we measured the orientation as I showed. And there's many other way of biasing trajectories for active particles in living system. There can be gradients of chemicals, of oxygen, of whatever. And we can use Phototaxis as a generic way of biasing the random walk. It can be illustrative of any other system. So with this simple characterization, I can show you one experiment where we coupled this behavior, this swimming behavior to a flow. And what we see, we called it photofocusing. So it's what you see in this movie. There's a millimetric channel here with the suspension, and we force a flow through the tube. And what you see at a certain time is that all the cells are getting focused in the center of the channel here spontaneously. And what we did here is that we use a light source upstream the channel. So the ingredients are all here. You have the cells that have their own swimming. We force a flow that is much stronger. I mean, the mean velocity is much larger than the swimming velocity. But still, there seems to be a coupling that orient the cells and make them focus on in the center of the channel in presence of light. So this is reminiscent of some older studies where a coupling between gravity and flow has been shown to lead to the same kind of phenomenon in the work of Kessler here. Or it's also at the base of some understanding of pattern formation that we can see in turbulent flow with the planktonic cells. So intuitively, you would say that in a turbulent flow, the swimming behavior of a cell is not relevant because the flow is so turbulent, so fast for them. But actually, it has been shown that the activity or swimming activity of planktonic cells coupled to a turbulent flow to lead to pattern formation as simulated in this work, for instance. So the mechanism is quite simple to understand. If you consider a particle that is in the bottom part of the channel, for instance, it has a preferred orientation toward light. Even though it's transported by a flow, it would like to orient its direction toward the light that is on the left. And meanwhile, it's subjected to the vorticity of the flow. As you have a quasi-flow, the velocity below the particle and on top of the particle is shearing the particle and giving rise to a vorticity torque. And those two simple ingredients together would lead to a migration because there's a swimming velocity to the center of the pipe here. So this is migration across streamlines in low Reynolds number is usually not possible with a passive particle. It's really because of the velocity the particle carries that the migration is possible. Yes? Yeah, because it's exactly the same ingredient, the same reasoning. If you put the experimentally, just that you observe a blurry, because the pipe is not 2D, they all go along the wall. So what you observe in the center of the channel is blur. But indeed, they go to the walls if you put the light downstream with the same ingredients. So to model this, we use a simple statistical, Eric Bertin developed a statistical description with the ingredients I gave to him, which is okay. I have put a parabolic flow that I know exactly the parameters. We know the mean velocity of the suspension, constant velocity of swimming, and the particles are subjected to vorticity reorientations due to the flow. So the orientation of the active particles are subjected to this vorticity, and here we have put an adjustable parameters as if the particle would resist a bit the vorticity of the flow. So a passive particle would just rotate with the vorticity given by the flow. Here we put an adjustable parameter, and on the right, so this is to predict just the probability for a swimmer to be at a given position. And on the other side, we put the random reorientation that we know of, and the direction distribution that we measured experimentally also. That is the direction toward light distribution of the suspension. The fact that we see a non-zero thickness of this focused region is due mainly to this distribution or this noise in the distribution. And with this, I will not enter the details, but we can quantify in quite a good agreement all the measurement that we can do in this experiment. We can measure the density of cells across the channel. This is the first panel. We can measure the length or the time equivalency in another referencer that the focusing would take, let's say, to be stationary. We can measure the width of the focusing that depends on the distribution. And all this fits remarkably well with a single adjustable parameter that I told you about, this beta, that actually we put it here because of a previous experiment that showed that an active suspension, so I will not detail this here, an active suspension of cooler type microswimmer, we showed experimentally that this increased the effective viscosity of the suspension. And this you can see as an extra stress or a resistance, like extra viscosity, let's say, of the effect of activity on the particle coupled to the flow. So this was nice to back up this hypothesis we had before. Now I just want to show that if I retain the ingredients of the model, here you see parabolic flow, constant swimming, etc. You don't see the specificity of the system. Only the numerical parameters are due to our experimental system, but this is quite a generic model that you can expect to apply to very different systems. in this way, I think managed to answer a bit the way I introduced the talk by saying that we are looking for generic behavior using a specific model system. And for instance, there's a question in immune system. You might know that white blood cells are able to have a swimming state, and this is happening in a blood flow that is very strong. Is the motility of those cells have a relevant role in the immune system for the migration of those cells toward the walls or the target? So this is an example of probable application. Is it okay? So I go on with another part, just in the same spirit. If we want to increase a bit the complexity of the system and to try to go a little bit further in the understanding of this kind of system. Now we looked at the interaction between those cells with the complex environment, and complex here is a very regular system of pillar, microfabricated pillars. And again, we look at the suspension behavior in an average sense. So there's in the literature some questions about the interaction between active particles and walls. So as I showed you, the particles are creating an hydrodynamic flow around them. So as a first order intuitive thinking you can have is that this hydrodynamic flow will interact with the wall. Maybe you know about image technique, image model. I don't remember the name, but a wall can be seen as a mirror for the particle. So you can expect this hydrodynamic flow to interact back with the particle itself, and depending on the type of flow the particle creates, and depending on the shape of the obstacle, you expect different behavior. You can see orbiting of particles around a circular obstacle, for instance, due to this interaction. An experimental paper suggests in a seemingly contradictory manner that the interaction between a cell and a wall is steric, so it's really a short-range interaction, and the long-range hydrodynamic interaction is no more relevant. You can have a direct interaction of the cell flagella even with the wall. And with the same authors, I think another theoretical model seem to come back to hydrodynamic interaction as relevant for the interaction with the wall. So just to say that it's not obvious to predict what would drive the interaction in the system. So the experiment is simple. It's again simple microscopy and looking at the trajectories of suspension in between those pillars, and the size of the pillar is a little bit on the same order of magnitude as the persistence length of the swimmers, and we vary the distance between the pillars here. The first observation, when we look at the orientation of the swimmer in average, or the distribution of orientation, you can see this anisotropy of orientation. So, for instance, so the different color correspond to different interdistance pillars, and what you see is that the distribution is peaked along the direction. Let's say along the pillars or perpendicular to the pillars. So it seems that most of the orientation are in these alleys between the pillars. Oops. So sorry for this. Here what we proposed with Eric Bertin again is to model this particle again as a active Brownian particle with the dynamics that we know of in a free medium. And instead of trying to model the pillars as they are exactly, we model them as an effective medium that would just add a reorientation rate. Okay, so this would add a frequency rate of reorientation. And in this reorientation, we'll keep the anisotropy that we observed in the experiment. So we'll keep the symmetry of the network the network of pillar, and this flower-like curve shows you that we just add a high probability of reorientation in the diagonal direction this time. Okay. So again, with this and with this model, we can derive. Okay, we can first derive the fit curves that you probably have seen before, but we can have the probability of orientation of the cells that is fitting quite well the experimental curve. And from this adjustment, we extract one value of the anisotropy factor, which is the frequency, right? So here it's written here, the frequency rate that we add as the mark of the effective medium. And with this value, we can extract in the model the diffusivity of the suspension. So experimentally, we measured again the mean square displacement and extract the diffusivity, the effective diffusivity at long time scale. And here we show that the diffusivity as a function of the distance between the pillars is well described by this simple model that superimposes an active Brownian particle with an effective medium that would act on the reorientation dynamics. And here we had one hypothesis. So here we show that a dry description is enough to account for our observations. But we had one hypothesis is that we take a very symmetric anisotropy of the medium and effect on the reorientations. We can wonder if this hypothesis we had to add is due to hydrodynamic interaction in the system that would channel the trajectories along the alleys in the network. So this has been generalized to different symmetry of networks that are not only square-like networks. Okay. Maybe I will just finish by an ongoing project, just for a few minutes here. What you probably have heard about crowd dynamics and a lot of phenomenology that is quite intriguing. Here it's a very illustrative example where people are asked to exit a room. On the right side, they are asked to go as fast as possible to exit. And on the right side, they are asked to go normal velocity. And we can see at the end of the movie that the room is evacuated faster when the people are going slower. So this has been called the faster is slower effect, which seems paradoxical when observed like this, and has been shown to be quite a general phenomenon in many systems that different as granular or sheep on the right side, or even in simulations. And one way of increasing the evacuation process is also paradoxically to put an obstacle just in front of the door, so that you make the people contour the pillar. So the question here is that can we define a new class of models where, when considering wet systems, are the interaction, the hydrodynamic interaction in the system modify this phenomenology, or do we find the same phenomenology? So we use again our favorite system. We use Phototaxis to drive the particles, and we ask them gently to go through a door. So experimentally, it's not as easy as we can think of, not as easy as I thought. And the observable in this system is to look at the statistics of the time, the exit time after the door, for instance. There's nice challenges in particle tracking on the left side of the movie that we are tackling now with the deep learning approach. And I just show you very qualitative and preliminary results just to show you the way we describe it. You have a building up of a clog before the door that we can measure experimentally, and we can show sometimes the apparition of an arch, like in granular matter, that this is known to block a process of flow that forms force chains in this system. So we have the feeling that we found the same here. And then the statistics of exit times can show polar behaviors as in many systems. We are now trying to try to understand more quantitatively if the power law exponents have a physical meaning. So just to end up here, we managed to have regimes of very active suspensions, which we didn't think it was allowed by hydrodynamic interaction. So they go and go for a dense phase. There's nothing to prevent this. We observe the clogging events, the typical arch-like pattern. And we have first preliminary experiments that seem to show that the faster is slower effect is recovered in this system. Okay. With this, I want to mention the PhD students that worked with me. So Mathieu Martin on the right side of the picture, and Marvin Brancos-Mebruni were the two last PhD that contributed a lot with the experiment with me. And Eduardo Alalam is now second year PhD on the crowd motion project. I work in collaboration with mainly theoreticians. Philippe Pella in the lab is doing numerics. Eric Bertin is also in the lab in Grenoble doing the statistical models I showed you. Walter Zimmermann is doing Lattice-Boltzmann hydrodynamic simulations, and the crowd motion project is done in collaboration with Bertrand Maury and Sylvain Faure. It's written Orsay, but I wrote also NS. They're in the math department. I had a coffee with them just before the session today, so they're just across the road. I don't know if I'm late, or I don't know if I added this just to try to be to fit in the thematic of the day. This has nothing to do with what I said now, but just to have, look like I'm doing ecology, but I'm not. I recently developed a project that aims at following the populations of planktons in the alpine lakes. So the idea is to build up a low-cost microscope, high throughput quantitative imaging. This has been proposed by people from the oceanic community. So it's Plankton Planet and Stanford University. Maybe you might have heard about Manu Prakash, who already developed the foldoscope. So in this same idea, they propose to share the development of a low-cost microscope dedicated to follow planktonic cells. So I get inspired by their proposition and adapted it to alpine lakes. So the three part of this project is to offer like a pedagogical platform for students. So they build up the system. Ecology students in ecology are doing also participating in this project, and then we use this as a base to as a sensibilization of people about global warming and stuff. And the data that we produce are getting into a database for optimizing the machine learning identification, automatic identification of plankton. So this is a replication of the planktoscope adapted to the lakes. Thank you for your attention.
We have questions for Salima.
You said at the beginning that the cells are moving in 3D.
Mm-hmm.
That's every results.
So no, actually the flow focusing is a 3D measurement for the crowd dynamics. It's intrinsically a 2D problem usually because it's inspired by pedestrians. But mainly it's a 3D experiment. You don't have always to. I mean, you can use projection in microscopy, but here, for instance, for flow focusing, we did simulations using hydrodynamics, and we needed to introduce 3D hydrodynamics because it's a 3D experiment. Of course, I don't do a 3D imaging.
Thank you for the presentation. I was wondering for the photofocusing, when there is an increase of the density of the organisms in the middle, does it affect the rheological properties of the flow?
No. So we did. So what we expect theoretically is that because of what I said before, there should be a rheological effect and also hydrodynamic interactions that makes clusters of cells because of the hydrodynamic interactions. We showed this with full numerical simulation, but we never observed it. So I guess here the noise is too large because the simulation had no noise. The noise is too large to observe this kind of effect, and the density of the jet is not as high as what we measured experimentally for the rheology. In a rheological measurement, I went to biologically relevant concentration. So it's really a magma.
Any question? Is there any reason for the eye spot is on one side of
But you have okay. I'm not at all pertinent for biological questions. But once I describe the mechanism, it seems convenient to have it on the side because apparently it affects the symmetry of the strokes of the flagella. So there's really like the chirality in the system. They are called trans and cis flagella. I guess this is related, but I'm maybe a little bit.
I was also wondering, in these dense expansions, you have shadowing effect sometimes.
Yeah, for the photofocusing, no, because we are in 3D and it's not so dense. In the crowding effect, because we are in 2D, I think we don't see, because as you might have seen in the movie, you can see directed trajectories on the right side too. So they're not shadowing themselves, but we have other problems like photoshock behavior. I think because of the dense system, we have, I don't know if it's related to what Gilles presented before. I don't think it's the same time scale, but anyway, they're forming aggregate. We have other problems, but not shadowing. I think because of the 2D geometry, I guess. You can see.
Yeah, that was very nice. I was wondering about the archive that you see very dense, because for inert material, I understand. For active, I have more trouble. We can get away from it.
Yeah, what I don't understand, I didn't expect to find back the phenomenology of simple sand suspension, sand file. So for me, what I don't understand is that there's so many similar phenomenology, and it seems that it's not really the activity. So it's such a dense. We're at 60% 2D surface fraction, surfacic concentration. So I guess I'm not sure you can call it active, even though they're still existing. So I guess, so it's what we are looking at with Bertrand Maury, how to connect friction-based model to the experiment, and do we need actually really activity, hydrodynamics? It seems that hydrodynamics is not relevant, so it's so dense system. But yeah, then I don't guarantee that this is reproducible, the arch thing. Now we are running a more controlled experiment recently.
Okay, anyone else? Okay, so we can thank Salima again.
Thank you again.